Hello everyone. In this tutorial, we are going to talk about the last portion of our transgenic mice tutorial, which is how to make a knockout mice. So what is a knockout mice? I'm going to present you with a drawing that I made earlier. Let's say you have this mice. Let's just assume that this is a mouse. And this mouse contains a gene X, and you're trying to remove this gene X to see what's going to happen to the mice because you are trying to prove your hypothesis that the gene X here is involved in a specific disease. And this is how the gene X looks. It has some flanking region here with some repetitive sequence, and you have the actual gene here. This is the coding region of it. So in order for you to knock out this gene, which means that you want to make it inactive or you want to remove it, what you will do is that you design a targeting vector. So this targeting vector, it's going to look more or less the same as your gene that is inside the mice, but with one difference is that instead of this actual CDS coding region here, you're going to put an antibiotic resistant gene such as neomycin. And as soon as this is fine, what you will do next is isolate embryonic stem cells. And the reason why we work with embryonic stem cells is these are capable of becoming almost any type of cell in the body. You're going to transfect your embryonic stem cells with this vector. And what you are hoping to have here is some homologous recombination. So this is going to swap like this, and before you know it now, inside the mice, you are going to have something that looks like this now. Once you put it into a foster mother and so forth, the new gene is going to be inactive. It's still going to carry the flanking region as we said, but instead of having the CDS, all it's going to carry is this neomycin resistance, and as a result, there's no way that we are going to have an expression, and as a result, the gene is going to be inactive. So this is how we knock out a gene in a mouse. But let's look at a theoretical point of view and what does the literature say about it. Making this mouse a knockout mouse, for obviously it's going to require some expertise and a very good knowledge of the complete DNA sequence of your gene of interest, at least quite a significant portion of it, which means that this is not a screening technique. You're not trying to screen to actually find out the gene involved in the process. And according to this method, you are going to follow 5 steps. Some of them are quite familiar, as you can see here. If you watch our previous tutorial, you still have a chimeric mouse, wild type, and the full story down here. You need to construct a targeting vector. So you build the gene that I just showed you earlier with a neo-R resistant gene. Then you need to have the gene targeting in your embryo. So the embryonic stem cells, they need to carry the counterpart as well. Then you need to select for the targeted gene cell. That's the reason why we are actually inserting this neomycin resistant gene, because you need to make sure that the targeting vector was successfully transfected in your embryonic stem cells. Then from there as well, you need to make sure that it was stable and it was done correctly. Stable, and I'm gonna add here correct, that it was correctly done. Then once that is over, the rest is something that you're quite familiar with: introduction of the targeted embryonic cell into a mouse embryo and into a foster mother. Then you're going to analyze the chimeric mice and you're going to do the inbreeding. But just for your own understanding, at the end you need to have now not just a homozygous, but you need to have a full knockout mice. So that is one of the biggest key difference there. All right. So once we have this knowledge, why don't we just zoom in into the specific steps? Let me show you now a little bit of a zoomed in image where we are going to construct our vector. So what happens when you construct our vector? During the construction of a vector, what we say, you are going to have something like this that looks similar to your gene of interest, but somewhere in the middle, what you're going to do, you are going to insert your neomycin resistant gene. I'm gonna put neo-R, and this gene, of course, codes for neomycin phosphotransferase that is going to disrupt. And if you pay attention here, this is your target gene that you're going to find in the embryonic stem cell, and this is your vector construct here. So in this vector construct, as you see, our neomycin is present. So we're swapping the blue for this orange, but our reds are still staying the same, which are this flanking region as well here. So these reds here, they're still staying the same, and that's what's going to allow for a successful homologous recombination here. Then what you see here, that's why we say that the neo-R gene is actually flanked by DNA sequences from the gene of interest in both ends, the two ends. And this gene-specific section, it's going to serve as an area of recognition for homologous recombination when the chromosomes and all the region are doing the recombination process later on as the cells divide. Then, in addition to that, we put also in a targeting vector a genetically modified gene to induce a second marker at one end, not in both ends, at one end here. We add this TK gene. It stands for thymidine kinase from your herpes virus. So we are going to add another region here. We will put this TK gene. And the reason why we put this TK gene, the neo-R gene is going to tell us that my vector got in the mice successfully. So embryonic stem cells and mice. So this vector got into this embryonic stem cell in a successful manner. It was in. And how would you check it? You're going to take your embryonic stem cells, all of them like this. You're going to grow them in a solution rich in neomycin. And all the cells that are going to die means that they didn't carry the neo-R gene, and those that have survived, then you will know, okay, these ones were actually carrying the vector. But to know that your homologous recombination was successful, at the end, you should have a little bit of disruption on the other side with your TK gene here. So this TK gene must be disrupted for you to have a homologous recombination and to know that your integration was actually successful. Because if your integration was successful on this side, like this, let me just put another drawing here. On the side, after successful integration, there will be no room for this TK because it was stable. All you will have is the neo-R here, and you are going to have your flanking region. So that's what it's showing here. Successful integration. As you see here, we no longer have the TK. In fact, let me show you maybe in the red color. The side, as you can see, the TK is missing. So that was a successful recombination in the mouse chromosome. So what does that tell you? Since TK is a gene modified from thymidine kinase of an herpes virus, if we grow it in ganciclovir, right? If TK was present, the ganciclovir is going to react with this herpes virus since it's a gene for a disease and eventually kill our cell again. But if the TK is missing, when you put the ganciclovir, it's not going to do anything to your embryonic stem cells. As a result, our desired outcome, which is this one here, I'm going to put it here, desired outcome would be to have embryonic stem cells that are, one, resistant to neomycin because they carry the neomycin resistant gene, and they must also be resistant to ganciclovir because the TK is missing here. The TK gene is missing. So this is our desired outcome. But if the homologous recombination was a random integration, the TK gene is going to persist. If the TK gene persists, what you're going to find yourself with is an embryonic stem cells that can resist neomycin, but unfortunately they are going to be killed by ganciclovir because the TK gene, the thymidine kinase, is present. As a result, this drug is going to infect, it's going to actually kill the infected embryonic stem cells. So in other words, what you would notice here is that the TK, it's kind of like infects your embryonic stem cells with herpes virus. That's why ganciclovir is going to kill them. All right. So I hope that was helpful. Please feel free to again watch and rewatch this whole video again. But of course, once you have your desired outcome, which is this one here with a very stable integration, you're going to take those cells, inject and implant them into a foster mother, and have your chimeric F0, F1 until you land with a full knockout mouse stay. I hope that this was helpful. Again, feel free to watch this over and over again, replay this until you understand what exactly is going on. Thank you.